Hi there. In this video, we'll learn about meiosis, which is another type of cell division used to produce a specific set of cells, namely the gametes, the sex cells, sperm and eggs, that are required for sexual reproduction. We'll talk about the differences between meiosis and mitosis. We'll emphasize the key physical difference in these two ways of cell division, which is that in meiosis, homologous chromosomes pair up during metaphase 1, whereas in mitosis, homologous chromosomes are treated independently. Then we'll also show how homologous recombination is necessary for proper chromosome segregation and how it creates new genetic diversity. This is an electron micrograph of an egg surrounded by many sperm cells. Both the egg and each individual sperm have equal genetic component. What the egg has in far greater abundance is cytoplasm and all the organelles that are involved. And this sort of graphically makes the point about why mitochondrial genomes are inherited maternally. There's just much more
cellular material
there. So this is a table comparing differences between mitosis and meiosis. In mitosis, which we've just studied, when a cell divides, you get 2 cells, and in meiosis, you're going to produce 4 cells. In mitosis, the goal is to produce 2 cells that are identical. In meiosis, all 4 of the cells are completely different and unique genetically. There's no crossing over in mitosis. That's because you're producing identical cells, whereas in meiosis, it's a requirement. All cells undergo mitosis, whereas meiosis only occurs in the gonads for the production of gametes. This is involved with asexual reproduction of cells, and this is as a prelude to sexual fertilization, producing a new unique genome. And then highlighting in this last item the physical differences. On the first slide, we talked about homologous chromosomes pairing up here in metaphase 1, and then the centromeres will not split, but the homologous chromosomes will be separated during the first division, but then in the second division, this is more like mitosis. In mitosis, there's only one metaphase and one anaphase, and as we saw, sister chromatids split there. On this slide, we see ways in which meiosis enhances genetic diversity, creating cells with completely new, never before seen genomes. In this hypothetical example, there's a cell with 3 types of chromosomes. It's a normal diploid genome, meaning that there is 2 copies of each chromosome, one from maternal origin and one from paternal origin. There are slight differences between the 2 chromosomes because your mother and father have slightly different genetic characteristics. So then after undergoing meiosis, let's look for instance at this cell here. The brown chromosome and the green chromosome come from the mother, but the blue chromosome comes from the father. So this is a cell with a completely new genome. The same is true for each of these other 6. You can ignore these first 2, which are recapitulations of maternal and paternal chromosomes, and that wouldn't actually happen in real life. But here you can see anyway, just because of independent assortment of the maternal and paternal chromosomes, you're creating new genetic complements. However, in reality, focusing here on just the red and the blue chromosome, in no case will a cell ever inherit the entire red chromosome or the entire blue chromosome. What's going to happen is, what's essential to happen for meiosis to occur is a process called crossing over, that the red chromosomes and the blue chromosomes, when they're aligned in the first metaphase, will start swapping some parts. Here you can see a bit of this blue tail has been stuck onto the basically red chromosome and it's been exchanged, a translocation with the red tail on the blue chromosome. And then on the second anaphase, when you split these sister chromatids, you'll see that you have the red and the blue one here, the blue and the red copy coming from here, and then same for cells number 3 and 4. And here you have 4 cells, neither of which is entirely maternal or entirely paternal. And this is a simplification over here on this side. In fact, all of these would be mixtures. And in fact, in reality, it is very close approximation that each chromosome that you inherit will be almost 50-50 in terms of origin. that the crossing over is frequent enough to roughly balance the amount of genetic material in any particular zygote cell here. And of course, a key thing to notice is that in this case, you have a duplicated copy of the maternal chromosome and a duplicated copy of the paternal chromosome. These zygote cells only have a single copy of each chromosome. If we call this chromosome 1, here you have a duplicated copy of maternal chromosome 1 and a duplicated copy of paternal chromosome 1, and here you only have a single copy of chromosome 1, which is an admixture of the two. This slide tries to draw the correlation between the stages of reproduction and the status of the ploidy of the genome. Ploidy is a word for the amount of chromosomal material. Our somatic cells are all diploid, and they produce germline cells, of which the germ cells, or the gametes, are haploid. That's the egg and the sperm, not drawn to scale here. We saw how much larger the egg is. The fertilization of these two reproduces a diploid zygote, which can then give rise to another diploid organism producing diploid cells, and then creating a new completely unique set of germline cells for further opportunities for reproduction. Looking at making the comparison here, again, here are the diploid parents, mother and father. You produce haploid eggs, and this fusion will recreate the diploid status. So in somatic cells, we have 23 pairs of chromosomes. In the gametes, there are only 23 chromosomes, not 23 pairs. This is referred to as diploid. This is referred to as haploid. And you might notice a logical inconsistency there. Feel free to bring that up in class. I'd like to see if anybody can catch that. On this slide, it makes explicit about what is inherited in future generations. We sometimes worry about the mutations that can occur in a somatic cell, which can be completely deleterious to your health. It can cause a cancer or other disease. However, it's not passed on to the offspring, and so it will not affect future generations. So it's only mutations in the germline cells that are passed on to offspring. That being said, we often hear in the news media about certain things which exposure to pregnant women can cause birth defects or birth issues. It's actually even worse than that. Exposures to a pregnant mother can actually affect their grandkids. The mother has a developing fetus. The developing fetus has its already germ cells, which it's going to use to produce its children. So the exposure to a toxic chemical can affect the grandchildren phase. This has been proven experimentally in mice. And here we see some of the biochemical differences between meiosis and mitosis. Since we've studied mitosis, let's look at that. Here we have homologous chromosomes. Let's call this chromosome 1, your maternal copy and your paternal copy. Then during the S phase, your homologous chromosomes get duplicated, and now you have actually 4 copies of the chromosome, 2 from your mother and 2 from your father. Then during mitosis at metaphase, these chromosomes will line up independently, and these duplicated arms here, these are called sister chromatids. And now at anaphase, when the chromosomes are starting to be pulled to the separate spindle poles, the sister chromatids are sorted, and then you get 2 identical diploid daughter cells. Now, meiosis starts out the same way and it ends somewhat similarly, but what's new here is an extra metaphase and an extra anaphase. But starting from the beginning, we have the homologous chromosomes. The homologous chromosomes are then duplicated. Now they line up in metaphase 1, but now instead of aligning separately, one on top of each other, they're aligning next to each other, the homologous chromosomes. Crossing over will occur. Blue and red chromosome arms, both of the chromatids will exchange genetic material. Here only one is pictured. And then the duplicated homologous chromosomes will be sorted, or segregated rather, and then there is another metaphase, metaphase 2, where all the chromosomes are lining up independently, and now we're going to split sister chromatids and produce 4 cells. because I should have mentioned we've had one cell division producing two, and then each of these two cells will divide again, producing four cells, and they all four would be unique, although only two chromosomes are unique here. You actually have crossing over of all four sister chromatids. These gamete cells are all haploid. The book labels it here as meiosis 1 and meiosis 2, but it's all one meiotic division. And then this is making the key stage even more clear with more detail. This is mitosis over here, and this is meiosis over here. And that here you see P1 and M1, the homologous chromosomes, they're lining up independently for separation of the sister chromatids. Here in metaphase 1, the homologous chromosomes M1 and P1 line up together. They'll be crossing over, and then separation of entire chromosomes. Now again, it's kind of surprising. If you looked at a chromosome which recombination has occurred, you would might think it'd be more challenging to separate these chromosomes rather than chromosomes that are lined up neatly. In fact, if you do not cross over the chromosomes, you will not ever segregate the homologous chromosomes to new cells. Maybe not ever, but you're not supposed to. It's only after crossing over, when the cell has performed a tension test to make sure that they are in fact crossed over, that the machinery, the protease machinery, will start to cleave proteins which are holding them together, then allowing those to segregate. And finally, I'll just mention we saw homologous recombination when we talked about double-strand break repair, and that was the intermediate, the chiasma, that was called the Holiday junction, and it was the manner in which you resolved at DNA repair. You would resolve the crossing over this way, and although you cannot see it in this projection, in meiosis you resolve it splitting between the back and the front. The back has most gray and a little bit of a red arm there. And here we see the separation of the homologous chromosomes. The kinetochores of the two sister chromatids are fused and acting as one. And then here they start to separate. The next slide will focus on this particular chromosome. And here we have at the second metaphase, now this is lined up, and now these kinetochores will separate the sister chromatids, being distributed to unique cells. And this slide is why I retracted when I said never. Disjunctions, non-disjunctions, where the chromosomes don't properly separate. This is a major cause of birth defects, and this occurs in meiosis. This also occurs in mitosis. But this is a major source of trisomies and also a major cause of spontaneous miscarriages. On this slide I have links to two very short and informative videos, and they are both included on the edX webpage. Okay, thank you for listening.